Hello. This is the presentation of the DNA replication model. Using this model, I will demonstrate the different steps involved in DNA replication. There are two important things I must note before we continue. First thing is that the process of DNA replication is semi-conservative. DNA replication is semi-conservative because through each strand of DNA replication will undergo its process and we have a resulting two daughter strands. Each daughter strand will have one strand of the original genetic material and the other complementary strand will be a copied strand from the original genetic material. So through each genetic daughter strand, a half of the strand is from the original genetic material. This way DNA replication is semi-conservative. Another important fact to know is that there is a difference between the two strands of DNA seen here and the process through which each strand is replicated. This yellow strand, the top strand, will be used in the demonstration as the leading strand, and this bottom strand, or the orange strand, will be used as the lagging strand. Now, it's important to note the differences in these two strands because the way they replicate are both different. This leading strand goes in the 3' to 5' direction. What makes this important is that DNA polymerase, which adds the DNA nucleotides to new strands, only reads in the 5' to 3' direction. So in the case of the leading strand, DNA polymerase will read across continuously with no problem. However, for the lagging strand, it actually is 5' to 3', which means that DNA polymerase cannot read continuously as it does for the leading strand. It actually has to read in short segments. It does a segment here, goes on to the next segment, and goes on. And through these short segments of DNA, we have Okazaki fragments, which is this fragmented way through which a DNA polymerase copies new nucleotides onto the lagging strand. So I would demonstrate how each strand copies independently of each other, but although I'll be doing it separately, in actual DNA replication they occur at the same time simultaneously. So without further ado, let's begin. The first step in DNA replication involves the unwinding of the double helix. For this, we have the enzyme known as helicase. Helicase is the enzyme that has a job of unwinding this DNA strand. So what it will do is that as it goes along the strand, the strands will slowly separate from each other and unwind. Although I did it continuously, in reality, helicase will slowly unwind one nucleotide at a time. So for this strand, it will unwind, separate a little, then goes on to the next one and separates, and so on and so forth. And it's also important to know that while these strands are separating, in front of helicase is tropoisomerase. Tropoisomerase has the job of making sure that DNA does not overwind as helicase moves through. And what's important to know also is that as helicase unwinds, we have a Y-shaped structure from this DNA known as a replication fork. This is essentially where the replication of the copied strands will begin from the leading and lagging strands. Beginning with the leading strand, we have the unwinded strand here. And what's important to note is that once it's unwinded, proteins called single-stranded binding proteins will attach to the strand and help stabilize it during replication. This is so it doesn't cause any problems, and so the strand stays mainly in place, allowing for an easy replication. So in order to fully begin replication for the leading strand, as I said before, it will continuously replicate DNA nucleotides from here to here because of the way DNA polymerase reads. So it will go towards the replication fork without a hitch, which is why it's called the leading strand. So the way to begin is that we have an enzyme called primase. Primase will add short RNA nucleotides to fully begin the process of DNA replication for the leading strand. So primase will attach to a nucleotide, and an RNA nucleotide will then attach to it from here. Primase will then go on to the next nucleotide, and it will add another RNA nucleotide from there. And this continues so on and so forth until DNA polymerase III is ready to do its job. DNA polymerase III, shown here, will attach and add DNA nucleotides to the strand. So as you see, it will go to the cytosine here, and we would then add a DNA nucleotide to begin replication. Now it's important to note that while there are 3 phosphates attached to this nucleotide, once DNA polymerase III does its job and attaches the nucleotides, 2 phosphates will break off as a release of energy to form this bond. It's an exothermic release of energy due to the formation of bonds. So while there are 3 phosphates here, there will only be 1 phosphate here. And then DNA polymerase will continue this through the strand, and we do this to the adenine. We add a thymine, which will catalyze the release of 2 phosphates, making a single phosphate here, and so on and so forth. So this will produce new DNA nucleotides down the strand. However, a different DNA polymerase, DNA polymerase I, will ensure that these RNA nucleotides from the primer will be removed so that the DNA nucleotides can be fully added on. So DNA polymerase will remove these primers. So first we'll do it for the cytosine. So guanine will detach from the RNA nucleotide, and we will then add guanine from the DNA nucleotide, once again breaking off 2 phosphates, and it would do it once again with the thymine, adding on a DNA adenine. So essentially this is how the DNA will replicate, so on and so forth, down the leading strand. Now the lagging strand is a different process, but there's some similarities between it and the leading strand. Now, when DNA replication occurs in the lagging strand, there are some differences that we had to note from the leading strand. While the leading strand was replicated continuously in the 5' to 3' direction, this cannot happen in the lagging strand because it runs in the opposite direction as opposed to the leading strand from 5' to 3'. So DNA polymerase cannot work towards the replication fork continuously. In fact, assuming our replication fork is here, DNA polymerase will work in the opposite direction away and actually replicate the lagging strand in short segments. Overall, it begin in the same process though. These single-stranded binding proteins will attach to the lagging strand to help stabilize it during replication. And then the process begins. Unlike adding RNA nucleotides and RNA primer through the use of primase here and going down, we'll begin somewhere in a segment, let's say here. So primase will come here, and you go add RNA nucleotides through primers through downstrand for a short segment. So first couple would be here. And then DNA polymerase III will then work to add DNA nucleotides for the short segment. Not the rest of the strand, but as a short segment. So DNA polymerase will attach here to the thymine, and and then the adenine is added here. Once again, which involves the breaking off of 2 phosphates. That's the next thermic release of energy. Then it will go to the guanine, and similar process shown from the leaking strand is also done here. However, this is a short segment. DNA polymerase 1 will also then work to remove these RNA nucleotides, which then adds DNA nucleotides. So from here, we have a short segment on the lagging strand of DNA nucleotides. This is known as an Okazaki fragment. And essentially, the leading strand is composed of multiple Okazaki fragments, which are just short segments of the DNA nucleotides that have been attached to the lagging strand. And one enzyme that is involved in the lagging strand that is not in the leading strand is this, DNA ligase. Assuming we have these 2 separate segments of cytosine, guanine, adenine, thymine, then cytosine, guanine here. Assuming we have these 2 separate Okazaki fragments, DNA ligase will come through and combine these 2 Okazaki fragments. And just like that, the rest of the lagging strand will be almost glued together through the use of ligase. And through this way, DNA replication is done. Thank you, and have a nice day.